It's such a pleasure to be with like-minded people and spend a morning listening to all of the amazing talks. So I think this particular talk is going to pick up on some of the themes, particularly from David Moira and also Ian latterly. But I'm going to be working with Ali, who'll be coming on immediately after me, and we're going to be working together to offer metabolic mental health coaching, and that's why we're going to be speaking together. I just really wanted to talk about the gut-brain connection, because there's been more and more in the way of research coming out, particularly over the last 10 to 20 years, to advance our understanding of the gut-brain connection and the relationship that that plays in relation to mental health. So it's going to be a whistle-stop tour because we don't have much time, so 15 minutes probably can't do the whole topic justice, but I'll do my best to give you an introduction. And I just really want to talk about some of the connections between those two different organs, but also the similarities, because when you look at it, you wouldn't necessarily think that the brain and the gut have an awful lot in common, but there are some quite obvious similarities when we go into it. Touch on the role that the gut has in regulating neuroinflammation, so inflammation in the brain, and also cover some of the clues from the research literature. So I'm glad I'm not the only one who has a slide with pictures of cells this morning. But I just really want to draw your attention to the diagram on the right. So that's a depiction of two gut epithelial cells. So in our gut, the gut is lined by a single cell layer throughout the gut of epithelial cells. And it's really it's an absolutely amazing organ when you think about it in terms of just how thin that layer must be and the great many functions that it serves. So I want to draw your attention to this is where I need the pointer, but the gap between the two cells, so that's known as a tight junction. And the purpose of the tight junction really is to regulate what passes through the gut wall into the systemic bloodstream and circulation. So if you go to the very top of the slide with the finger-like projections, those are the villi within the gut, and they project into the middle of the gut essentially. And you can see from the depiction the different microbes that occupy our gut, often referred to as the gut microbiota or gut microbiome. And then coming down to the opposite end of the picture, you can see the bloodstream. And there are also many immune cells depicted there. So a glial cell, for example, is part of our immune system. And there's a lot of crosstalk that happens between the immune system and our gut because of their close proximity. Something that seems to be coming out more and more in the way of research, particularly in relation to chronic disease and illness, relates to something called leaky gut or increased intestinal permeability. And that's going to be a big focus of the topic just in this brief talk. But I'd like you also to notice the two words on the screen, claudin and occludin, because these are proteins that are extremely important in terms of regulating the tight junctions and regulating the performance of the tight junction in keeping toxic molecules out and toxic molecules kind of within the gut and preventing them from crossing over into the bloodstream. And there are various other molecules depicted there, so zonula occludin as well as another protein involved in regulation of the tight junction and its function. There are lots of things that can cause leaky gut or increased intestinal permeability, so it's not just diet. Stress is one that we know can affect gut permeability. Also alcohol can. But often when I'm asked to speak about the gut or think about the gut, the first thing I think of is gluten. So it was interesting just to pick up on your question from earlier. So the zonulin-gluten interaction is very well researched and very well written about in the literature. And we know it's not just people with celiac disease that this affects. So gliadin is a component of wheat protein, and we know that it interacts directly with the zonulin within the gut epithelial barrier. And it binds to it essentially, and opens up the tight junctions and directly causes leaky gut. And the difficulty, which I'll come on to in later slides, is that when you have a leaky gut, then there are all sorts of things that can pass across from the gut into your bloodstream that have quite a detrimental effect elsewhere in the body, particularly in relation to causing inflammation and immune system activation. I won't spend too long on this, but the microbiome, again, what I think is a really fascinating subject. And I think there's a lot that we still don't know about the microbiome. But what we do know is that within our gut is resident to many different microbes of different types, so viruses and fungi and protozoa, but the vast majority are bacteria of all different types and strains. The actual DNA, so genetic component of the gut, far surpasses the amount of DNA in our own human cells. So that just gives you a bit of an idea. And actually our gut is the biggest interface that we have in the body with our outside world or outside environment. And the microbiome fulfill many different functions. I just really, for interest of time, want to draw your attention to the top left in the diagram, which speaks about modulation of the central nervous system. And so there are various different communication pathways between the gut and the brain. There's a hardwired connection, which is the vagus nerve, but there are also other neurological connections, particularly in terms of the enteric nervous system that resides within the gut. So signaling between the gut and the brain and vice versa can happen along the neurological connections, but it also happens along more chemical type connections. So our hormonal system, or endocrine system, so it's particularly cortisol that's involved in signaling between the two organs, and also the immune system and cytokines. So cytokines are not always, but tend to be pro-inflammatory immune mediators that are released by the body in response to a stimulus, and we do know that they are heavily involved in the signaling that happens between the gut microbiome and the brain. And something just to say is that there tends to be bidirectional communication between the two organs. So we know that gut communicates with the brain and brain communicates with the gut. So I just wanted to show a couple of examples of that. So one example of the gut communicating with the brain is that of short-chain fatty acids. So we know that bacteria in the gut produce short-chain fatty acids, and that they can also influence serotonin release, but also influence memory and learning processes in the body. And then another example is that this was a mice study, I think, or it was an animal study, certainly of some sort from memory. And researchers were able to show that even a relatively brief stressor, so you could think a kind of psychological type stressor, was able to induce demonstrable changes in the composition of the gut microbiome. And that was a relatively brief stressor of just 2 hours. So the obvious question is, how could this be if there wasn't any connection between the two organs? Now, just coming to some of the similarities, something we have in the brain, or kind of surrounding the brain really, is something called the blood-brain barrier. And it's part of the systemic circulation, so it's blood vessels essentially that make up this part of the body. And its main purpose is to regulate nutrient diffusion and nutrient access to the brain, but also really to keep out noxious substances and keep out toxins from the brain from entering. And we can see there are some similarities between the endothelial cells and the blood-brain barrier. So it's epithelial cells in the gut, but endothelial cells in the blood-brain barrier. So they're not exactly the same, but they are also a single cell layer with tight junctions. And it's also the same proteins that regulate the tight junction function in the blood-brain barrier as in the gut. So a lot of the same genes that encode for these proteins, in theory, it looks like can actually affect the permeability of your blood-brain barrier as well as your intestinal permeability. And so this leads on more to the connection between leaky gut and leaky brain, as I like to call it. So one of the real difficulties when you have increased intestinal permeability is that you can have leakage of less than ideal substances from the gut lumen across into your bloodstream. And then these substances can travel far and wide in the body, far and wide from the gut. And one particular area of research that is very well documented is that of LPS, or lipopolysaccharides. So lipopolysaccharides are a component of gram-negative bacterial cell walls. So they're just a component of bacteria that is considered an endotoxin when it enters into your bloodstream. And so you can see from the diagram top left, we know from research that having just these LPS in your bloodstream can lead directly to activation of microglia, which are resident immune cells within your brain. And when microglia become activated, they then release further pro-inflammatory cytokines or pro-inflammatory immune mediators that fuel a self-fulfilling cycle of further inflammation. Oxidative stress and mitochondrial dysfunction also comes into this, because when microglia become activated, they produce free radicals such as superoxide, which you might not be able to read, but it's depicted on the bottom arrow there. And essentially when microglia become activated, this contributes to the genesis of neuroinflammation, so inflammation within the brain. And it can lead on to neuronal cell death and damage, so death of nerve cells within the brain. There are other, so I mentioned about astrocytes, they're another type of immune cell in the brain that can also become reactive. And one thing I should have said was that when LPS translocate into your bloodstream, we know that it triggers off systemic inflammation in the form of production of pro-inflammatory cytokines, these immune mediators. And you can have elevation of different ones, such as tumor necrosis factor alpha and different interleukins, and you just get into a very vicious cycle, ultimately resulting in inflammation. And this is just another clip from a study. It was a really nice write-up of the different mechanisms involved in neuroinflammation and depression. So we know that probably about a third of people with depression have elevated inflammatory markers. So it's not everybody, but there's certainly a significant proportion. And you can see from the diagram just the different interactions between the gut bacteria, releasing LPS that can then go on to activate microglia, but these LPS have also been found in close proximity to amyloid plaques and neurofibrillary tangles that happen in different dementias in the brain. And yeah, there was a piece in the so the researchers say microglial activation appears to be a key event that triggers changes in signaling cascades and gene expression that would be determinant for the onset of depressive symptoms. So this was a really recent study that I like. So this was in humans. A lot of the research is in animals, but there are some human studies that we have now. And they compared 50 people, adolescents actually, so young people with depression, a diagnosis of major depression with healthy controls. And the researchers measured markers of intestinal permeability and blood-brain barrier permeability. So they tested claudin-5, which is one of the main proteins involved in regulating the tight junctions between cells in both the blood-brain barrier and the intestine, zonulin and fatty acid binding protein, and LPS again features here. And what they found was that levels of these different markers were significantly elevated in the cohort that had depression when compared to healthy controls. And so they concluded by saying that their findings advance our understanding of the pathophysiology of intestinal barrier injury, bacterial translocation, and blood-brain barrier injury involved in adolescents with major depressive disorder. We have some other clues from the research. So we do have case reports of, just touching on your point earlier, of gluten-induced psychotic episodes. So there was a nice one written up in a 14-year-old girl, and the researchers did a double-blind test. So somebody at age 14 whose psychotic symptoms resolved when going on a gluten-free diet, they were able to test using wheat powder, I think it was in pill form, and then rice powder in another pill form, and they were able to show for this particular individual that psychotic symptoms did not recur when the patient was given the rice pill, but when they were given the wheat pill, they did recur, and all the symptoms resolved on a gluten-free diet for that particular individual. But anyway, coming back to other aspects of the microbiome, so there are case reports of clear psychotic episodes having been brought on by broad-spectrum antibiotics. So ciprofloxacin and cotrimoxazole are both broad-spectrum, and I think it's a safe assumption that they would have quite an antibacterial effect within the gut. I think researchers kind of postulated that there may have been some effect on glutamate when it comes to ciprofloxacin, but really they don't really know otherwise. And I think with the increasing research that's coming forward, it seems a safe assumption to me that the microbiome may have had something to do with those particular cases. Another human study where they took a sample of deceased people, obviously, and examined their brain tissue. And most of the subjects in this study had autistic spectrum disorder, but there were a few in the study who also had schizophrenia. And they had slightly different results between the two. But what they found essentially was that there was different expression of these different tight junction proteins in the sample with autistic spectrum disorder. So they conclude there's altered expression of genes associated with blood-brain barrier integrity, coupled with increased neuroinflammation and possibly impaired gut barrier integrity. So they said 3/4 of the ASD sample had reduced expression of barrier-forming tight junction components, as well as other changes in different inflammatory markers and proteins involved in regulating both blood-brain barrier and the intestinal barrier. We also have studies in healthy people showing that taking a probiotic, a mixed probiotic, can lead to lowering of subjective levels of distress and anxiety. But I wouldn't necessarily rush out and say to people to just take probiotics because I think it's very complicated and there's a lot of contradictory results in the literature out there. And the microbiome is a very individual thing. Anyway, in the interest of time, we'll move on. And then there's also there's a few functional MRI studies. So functional MRIs tend to look at blood flow within the brain in real time. And what this particular study was able to show was that supplementing people with a probiotic and a fermented milk product actually led to changes in the people's brains that showed up in the scan, particularly in the regions that deal with emotion and sensation. Coming back to this LPS, so the endotoxins that come from the gram-negative bacteria in the gut. And this was another study, a bit older, where they looked at 51 study subjects, 28 with major depression, 23 controls. They measured antibodies to the LPS and to some of the other gut bacteria. And they showed that there was quite a significant antibody response to the LPS in those subjects with depression, which they conclude indicated gut permeability issues in that cohort. It's actually interesting, there are some other studies in humans showing that if you inject healthy people with LPS into their bloodstream, within 2 hours they develop a full house of depressive symptoms. So depressed mood, anxiety, anhedonia, or loss of enjoyment, reduced energy, reduced motivation, and those effects can continue for about 4 or 5 hours. The lipopolysaccharides, so it's a component of the cell wall of gram-negative bacteria found in the gut. So really, I'm going to need to let Ali speak in a second. I think the missing links in terms of mental health and how this can be affected are really come down to inflammation. And when we talk about peripheral inflammation in the body, immune system activation is a big part of that. So the role of these pro-inflammatory cytokines and what they play. And we know that cytokines in themselves can cross the blood-brain barrier and enter the brain, and they can also contribute to a leaky blood-brain barrier or increased permeability of that. And once you have activated microglial cells in the brain, you just go on to produce more and more inflammatory mediators, and it can also result in glutamatergic excitotoxicity, so one of the main excitatory neurotransmitters in the brain, and that Ian had mentioned in his talk. And I'd be absolutely remiss if I didn't mention the ketogenic diet somewhere in this short talk. So this was an animal study again, but it was able to demonstrate that the ketogenic diet mediated microglial activation, and it says restores microglial activation here, but there's actually 2 different forms of activation. One is a good form, and then one is a neurotoxic form. So the ketogenic diet was able to alter via the beta hydroxybutyrate, so the main ketone body, it was able to alter how these microglial cells were expressed to put them into a more sort of calm, anti-inflammatory state. And I won't take credit for this poem, but I have a friend who's very scientific and very into the gut-brain axis, so I thought I'd leave you with a poem that they wrote for me, because they said everybody likes stuff like this. And